Hello students, welcome to the theory class of Principles of Plant Breeding, PSH 123. This topic is on marker-assisted selection. To start with, let's discuss what is a marker. Genetic markers are the biological features that are determined by allelic forms of gene or genetic loci and can be transmitted from one generation to another. This means it can be any biological features. It may be in the form of morphological trait, or it may be in the form of chromosomal trait, or at the level of DNA, which is associated with the different forms of alleles produced by the genes. Okay, we have already learned that genes have different forms. Okay, so because of the according to the different forms of genes, if there is different polymorphs of DNA, chromosome, or different morphological characters, and which can be transmitted from one generation to another, which differential polymorph can be transmitted from one generation to another. This can be used as a genetic marker which follows a Mendelian inheritance. So next coming here is the classification of genetic markers. We can have two broad categories of genetic markers. First one is classical marker and DNA markers. Classical markers are those type of markers which were used before DNA markers were discovered in 1980s. And classical markers can be further classified into morphological markers, cytological markers, and biochemical markers. DNA markers can be further classified into hybridization based markers and PCR based markers. So let's see what are the different types of markers here. First one, morphological marker. So considering the different traits which are studied in Mendelian inheritance as an example, so we have different kinds of traits, 7 different kinds of traits. Say for example, tallness and dwarfness, purple color and white color of flower. So if it is at certain times, if it is correlated with the yield of a crop plant, so it can be used as a marker. Say for example, tallness of a plant type is correlated with high yield, and dwarf plant type is correlated with low yield. You can select high yielding plants by selecting the tall type. You can predict the high yielding plant type by looking at the tall plant type during the vegetative stage only before flowering. And if you have to actually wait for the actual crop yield, it will require the whole maturity period. But instead of that, you can just select from the correlated trait which is associated with the main economic yield. So this is one of the morphological character. This is not only so, you can find various other morphological characters in various other crop plants, like stem pigmentation in bendy, and so and so. And some of the demerits which are associated with such type of markers are the availability is very limited, and many of these markers are not associated with important economic characters which are referred here as such. Okay, next one is cytological markers. Any type of markers which is associated with chromosome, at the level of chromosome. Okay, it may be differences in size, maybe differences in shape, deformity, segment of chromosome, bending pattern, or increase or decrease, deletion or addition of a particular chromosome. Okay, so in this case you can see two different chromosomes, two different karyotypes of two individuals, which are one being normal human and other being Down syndrome, one suffering from Down syndrome. So we can have a clear differential picture of one having Down syndrome having an extra chromosome 21, and which is not there in 21 normal human chromosome. So such type of changes in the karyotype or in the structural differences, numerical differences can be seen, which can represent the differential phenotype. It means diseased individual and the normal individuals can be distinguished by this presence or absence of this particular segment. This is not only so, it can also be represented by bending patterns, so and so. This is just an example. Another type of cytological marker can be detection of an introgress gene into an individual with the use of fluorescent level probe. That is called as fluorescent in situ hybridization, FISH. So such type of marker has some demerits. Say for example, low polymorphism, the amount of polymorph present here are very less, and needs special techniques. Say for example, fluorescence in situ hybridization technique is high cost and very it needs it is a different types of special techniques. So it comes with high cost. Next one is biochemical markers. These are isozymes, allozymes, or different proteins which are produced by different alleles, which are produced by a single gene. Okay, just remember that a gene has different alleles. Okay, for the different allele, if the different alleles responds to different production of different isozymes, it it confers to, it can act as a biochemical markers. What are isozymes? These are multimolecular forms. Okay, isozymes, different isozymes are having different morphological features but having similar functions. Okay, and because of the different molecular structure, they will move at a differential speed in the gel, electrophoretic gel. So we have here some of the some of the isozymes which are having highly branched, and some of the isozymes which are having low branch. So having large amount of surface area reduces the speed of the movement of the particular isozyme in the gel pick. Okay, in the electrophoretic gel. So that way you can identify individuals, you can identify the isozymes based on the differential speed of the movement of the in the gel pick. Okay, some of the demerits here are there is limited in number, and the amount of polymorphism which you can see here is very less as compared to DNA markers, and it is usually affected by plant tissues and different plant growth stages. Okay, so some of the isozymes, some of the enzymes produced are very limited to only certain stages. Okay, either it may be at the filling stage, or it may be in the stage of grain at the flowering stage. Okay, some of the enzymes are produced at the maturity stage, so it differs from time to time and there is a limitation for that. Next one is DNA marker. So this is another broad category of genetic marker and very much essential for marker assisted selection. So the word molecular marker directly correlates with DNA markers, rather it should also consider biochemical marker, because biochemical marker considers protein which is a part of molecule. Okay, so but molecular, when it comes to the term molecular marker, it is directly correlated with DNA markers. Okay, in some books the molecular marker is classified as biochemical marker, DNA markers, but molecular markers are directly correlated to DNA markers. Okay, and what is a DNA marker? It is a small region of DNA showing sequence polymorphism in different individuals within a species or among different species. What does it say? If for a particular species, say for example pea plant, if there are several varieties of pea plant having different alleles of it, okay, A, B, C, and A small a, such and such, so they will produce different DNA bands corresponding to different alleles which is produced by the particular, which is present in a particular crop species. Okay, so that means DNA band, you can detect polymorphism at the level of DNA variation in the DNA sequence. Okay, next one is type of DNA markers. So some of the DNA markers given here are restriction fragment length polymorphism, amplified fragment length polymorphism, random amplified polymorphic DNA, variable number of tandem repeats, microsatellites or simple sequence repeats, single nucleotide polymorphism, short tandem repeats, so and so. There are various other markers also, but we are only going to discuss only few of them because this is a chapter of marker-assisted selection. We'll focus on marker-assisted selection. We'll know the mechanism, how the DNA marker works, and we'll move on to marker-assisted selection technique. So first of all, let's discuss what is RFLP marker, which is the first kind of marker which is discovered in 1980s. So what we do here, the DNA molecule of say for example individual A and B is cleaved by the same restriction enzyme. Individual A and B are from the same individual, from the same organism. Say for example pea plant, okay, two pea varieties A and B where DNA molecules or the genome is cleaved with the same restriction enzymes EcoR1. Okay, it gives restriction fragments. If in case of individual B also, if it is cleaved, it will give the same restriction and restriction fragments if there is no mutation. But what happens in case of mutation? Okay, suppose these are two individuals and these are three restriction sites. If there is a mutation which destroys the restriction sites of the particular DNA segment of, say for example, the individual B, so there will be no cleavage of this fragment, which gives in the gel picture what will be there, it will give a loss of fragment in the gel picture. Okay, so detection of polymorphism in the differential fragments gives give two different markers, RFLP markers, which can detect allele A and B, presence of allele A and B in the in the genome due to mutation. Next one is simple sequence repeats. So in case of a particular individual, say for example human being, most of the DNA contained in the genome of human are non-coding sequences, and only few, which is represented here by blue color, is coding region, which gives all the metabolic activities, which codes for all the protein functions for metabolic activities of the human body. All these are junk DNAs which doesn't have any function for protein. And among these junk DNAs, some are repetitive DNA. Okay, we can use this repetitive DNA for plant breeding. We can use this as a marker to tag along with some of the useful gene of interest. So how can we use this? It is usually scattered throughout the chromosome of, or throughout the genome of the individuals. Okay, so we can, if there is a gene of interest here, so we can tag along with any of these markers, or any of the marker which is nearby to it, or flanking markers which is bordering the gene of interest. If it is here, so you can tag along this particular marker. Okay, and then you can also use the markers if necessary, if you need to tag along with any of the gene of interest which is present here or there. Okay, next one is the type of, what is a simple sequence repeats. It is a repeat of a simple sequence of nucleotides, say for example GATA in this case. So we have here three individual genome, 1, 2 and 3. So in this we have repeat differences in repeats of GATA. The first two individuals having 8 repeats of GATA, while the third one is having 9 repeats of GATA. This so happens because of mutations on the way due to replication, and first two individual indicates presence of same type of allele, capital A, while the third genome indicates small a. Okay, if we take it as an example, so differences in the alleles capital A and small a can be detected by differential repeat sequences, repeat number 8 and 9. So it can act as a valuable marker for detection of particular gene of interest. So what is a microsatellite? Microsatellite markers are usually considered for those type of markers where the repeat sequence is from 2 to 5 base pair sequence. Okay, and if it is 50 to if it is 10 to 50 base pair sequence, it is known as mini satellites. Okay, next one is single nucleotide polymorphism. So in individual, if the individual varies only for a single nucleotide, and if it gives particular changes in the morphological character, you can consider that as a single nucleotide polymorphism. So you can see here changes in different individuals, changes in individuals based on the nucleotide sequences on only one nucleotide, in every individuals. Okay, and the possibilities are the presence of only four different types of nucleotides: A, G, T, and C. Okay, so for this reason, the chances of the number of polymorphism in SNP marker is a bit lesser than SSR marker, which in case of SSR marker, what will happen, that there can be various kinds of repeats, like 2 repeats, 3 repeats, 4 repeats, 5 repeats, so and so up to 50 repeats. Okay, but in case of SNP markers, there are only 4 different types of repeats possible. But if you repeat another one, A, G, T, and C, after A, G, T, and C, if you repeat another A, so it will be similar type. There won't be any polymorphism. So the level of polymorphism is less when it compares to SSR marker, but the chances of occurrence of these SNP markers throughout the abundance of SNP markers is quite huge, because the chances of occurrence of the polymorphism is huge, because it happens at the nucleotide level. Okay, taking sickle cell anemia as an example. So if there is a transversion mutation of T into A, so it will lead to transition into formation of RNA GAG and GUG. Instead of GAG, it will form GUG in sickle cell individuals, forming protein glutamic acid into valine. From normal, it will convert into valine. So what will happen in the morphological character? The normal sized RBCs will become sickle shaped RBCs. Okay, and because of these sickle shaped RBCs, it will block the arteries and thus producing the morphological character in a larger picture in the individual level. Okay, which is sickle cell anemia. So sickle cell anemia can be directly correlated to the changes in the nucleotide level, while normal individuals at the nucleotide A. Okay, so because of these changes, you can also consider the single nucleotide polymorphic markers as the marker you can tag along with any morphological changes in the character. So some of the attributes which confers for ideal markers. Some of the criteria for ideal markers are as such: easy and low cost of use and analysis. The ideal markers some of the ideal markers, it should be very easy to detect, and the cost involved of analysis of markers, PCR, DNA extraction should be lesser compared to others. And next one is small amount of DNA is required, because if large amount of DNA is required, you would have to extract DNA from time to time, and if you have to repeat the experiment, you have to extract the DNA from here and again. Okay, and which is difficult in case of non-cropping seasons. Next one is codominance type. If you remember clearly in the Mendelian laws of inheritance, the codominance in case of codominance, it means in Mendelian inheritance, it means the presence of the expression of both the alleles in a particular individual, in a heterozygous individual. Okay, in this case also it's nearly similar. In the gel pic, the DNA bands will show the DNA band of both the parental type. Okay, so for P1 and P2, if the DNA band is shown at higher these two level, and if the F1 shows at both the levels, so it will indicate a heterozygous type. And the advantage of this being, it can distinguish between a heterozygous type and a homozygous type. Okay, so you can see here a codominant marker in either indicate the presence of capital A, small a, which is here, and a heterozygous genotype. If it is a heterozygous genotype, capital A, small a, it will represent both the bands. While in case of dominant markers, it will represent only presence or absence. In heterozygous genotype also, it will represent only one single band. So this is the difference of codominant marker and dominant markers. Next one is repeatability or reproducibility of results. That means if you repeat the experiment in the next year or the next time, so it should show the same results. Okay. The next one is a high level of polymorphism. In case of SNP markers, the level of polymorphism is A, G, T, and C. 4 different polymorphism is possible, and which is compared to SSR marker, you have various kinds of polymorphisms, like from 1, 2, 3, 4, 5, 6, 7 up to 100 and more than that also. You can have various kinds of polymorphism when compared to SNP markers. So for an ideal marker, it should have a very high level of polymorphism. Okay. Next one is occurrence and even distribution genome wide. Okay. We're talking about SNP markers. The chances of occurrence of this mutation at the nucleotide level. So because of the presence of mutation at the nucleotide level, the abundance of SNP marker is quite huge in the genome. Okay, so even distribution of genome is quite high in SNP markers. So SNP is ideal for such a trait. Next one is marker assisted selection. What is marker-assisted selection? It refers to such a breeding procedure in which DNA marker detection and selection are integrated into a traditional breeding program. So it is an upgradation, or it is a biological tool which uses DNA marker in assisting the selection program of the individuals which are yielded by crossing two different individuals in up to generations. Okay. Looking at the technique of marker-assisted selection or marker-assisted breeding, if we have a cross between two different individuals, one being susceptible and having all the significant agronomic characters like yield, and one being resistant and having non-significant characters of yield, if we have a cross between these two, showing the different band patterns, okay, then it will produce F1, selfing F1, it will produce F2 generations. And remember, in case of marker-assisted breeding program, we have to choose from a large number of F2 populations. From F2 population, if we have to select, we have to conduct activities of marker-assisted selection, which is known as genotyping. Okay. Selection of plants based on the morphological character is known as phenotyping, but selection of plants based on DNA level is called genotyping. First of all, the tissue samples of the different plants of F2 is to be taken into account. So we have to collect sample and we have to extract DNA. Samples should be taken from such tissues, young leaves, and it should not be taken from twigs. And so if it has to be taken from twigs, you have to use liquid nitrogen, and then PCR has to be done to amplify that particular DNA segment of gene of interest. And next one, you have to go for gel electrophoresis. You have to run it in a gel and check for the marker amplification of marker, and after running it into the gel electrophoresis, you will be able to see a gel picture looking much similar to this. So when you compare the two parents and the F2 progenies of the gel picture, you can select those individuals which are having resistant type, okay, and avoid those individuals which are having susceptible type. Okay, avoid which are having susceptible plant type. So marker-assisted selection can be followed in this way: selection of plants on the basis of genotyping, on the basis of molecular markers. Okay. Next thing is comparison between a conventional backcross breeding program and a marker-assisted backcross breeding program. So in case of conventional backcross breeding program, we are going to cross between two parents, one recurrent and donor parent, and the F1 is to be continuously backcrossed into the recurrent parent in order to retain all the agronomic characters of the recurrent parent and attain only the part of the genome which is desired for transfer. Okay, so when in backcross breeding program, in conventional backcross breeding program, continuous round of backcrossing is required, while in case of Markerassisted backcrossing, only few backcross breeding program BC2 is required. The desired DNA can be selected in the one step only, few steps only. Okay, so if in certain cases, if there is a cross between P1 and P2, and if F1 is yielded, it has to be crossed with first of all with the recurrent parent, and our goal should be to attain as much amount of the recurrent parent and little amount of the donor parent which is having the desired gene. Okay, because most of the time this donor genome is associated with detrimental effect, and and it is also known as association of it is also known as genetic load, which means genetic load is the the amount of detrimental effect which is associated with the gene of interest transferred from the donor gene. So if you grow a large amount of BC2 generations, you can expect something like this, which is having recurrent genotype, large amount of recurrent genotype and small amount of the donor parent genotype, least amount of donor parent genotype, having all the advantages of agronomic characters of the P1 and the advantages of the donor parents. Okay, next one is during Markerassisted selection we follow three different types of selections. First of all, foreground selection, which is selection of the gene of interest which is transferred from the donor parent. Second one is a recombinant selection, which is selection of recombinant events between the gene of interest and the marker which flanks between the gene of interest and the markers. Okay, so this the use of recombinant selection is it reduces the amount of genetic load which is supposed to be there if there is association of, if there is tight linkage of this gene of interest with any detrimental gene which is from the donor parent. And next is background selection. So so this is one of the advantages of Markerassisted backcrossing. Recurrent selection can be avoided. Continuous round of recurrent selection can be avoided with by selecting markers which are not linked with the gene of interest but linked with the all other characters associated with all other genes associated with the recurrent parent. So we have three different types of selection: foreground selection, recombinant selection and background selection. And here are some of the advantages of Markerassisted selection over conventional plant breeding program. First of all, the plants can be screened or plant can be selected at the seedling stage or even before growing the seeds. You can extract DNA from seedling stage or seeds. Okay, so this avoids time consumptions. You can select plants based on the DNA, okay, or from DNA marker from the seedling stage itself. While in conventional plant breeding, you have to wait all the way to maturity to express the yield. And when in case of the second advantage, it is the difficult traits. Selection of difficult traits, expensive traits can be achieved. Say for example, polygenic traits, which is fluctuated by environmental effect, can be selected with the help of Markerassisted selection. It has a better chances of accuracy. Selection has a better chances of accuracy in Markerassisted selection for such a trait. And next one is for traits with complex inheritance. It also comes polygenic traits. It also comes to play in this, having high environmental effect, and those characters which are having low heritability and genetic advance. The next one is recessive genes, which can be maintained. Say for example, some of the desired character is controlled by recessive genes, so we can also track down, since it is having low heritability, and as homozygous and heterozygous plant can be distinguished between the head of codominant marker. So homozygous recessive can be identified, the presence of this type of genes can be identified with the help of codominant markers also. Next one is choice of parents in crossing program markers. Not only selection is done in F2 generation, you can also do selection for the parents in the backcross breeding program or in any plant breeding program, okay, with the use of markers. So there are some factors which which decides the accuracy of marker-assisted breeding program. First one is a genetic map which is adequately associated with uniformly spaced polymorphic markers, so as to map the gene of interest or QTL with the associated marker, okay. And next one is close linkage between the QTL or measured gene of interest with the adjacent markers. So a close association of as nearly as 5 centimorgan or lesser than that, which is known as tight linkage between the gene of interest and adjacent marker, is necessary for marker analysis. And next one is adequate recombination between the markers and rest of the genome, okay. So adequate recombination is supposed to mean recombinant events between the marker and the gene of interest, and it also is necessary for while doing QTL mapping or measure gene mapping, okay. And next one is ability to analyze a large number of plants in a time and cost effective manner. So this is one of the factor, okay. It has to be able to analyze a large number of plants in a very short period of time and very less amount of money. The last thing is application of marker-assisted selection in plant breeding. So one of the applications, it can be used in transferring resistance to biotic and abiotic stresses, disease resistance and abiotic stress resistance, say for example drought and cold tolerance, okay. So for biotic resistance, it is usually conferred by oligogenic genes, and for abiotic stresses, it is usually conferred by polygenic traits. Marker-assisted selection has been found to be effective in both quantitative and qualitative traits, so it can be equally effective in those biotic and abiotic stresses. It is useful in gene parameting for disease and insect resistance. What is gene parameting? It is the introgression of 2 or more different genes for resistance against 2 or 3 different disease, or 2 different strains of the particular disease or insects of the particular disease, okay. So or resistance against some of the disease and some of the insects, okay. So gene parameting is introgression of more than 1 gene into an individual, okay, or a single host. The next one is transfer of male sterility and photoperiod insensitivity. This you can see, these are the 2 important breeding objectives, okay. So male sterility is for mass multiplication or mass supply of seeds, mass production of seeds, and photoperiod insensitivity is for those for crop plants where long day period of sunlight is required for high yield, okay. And next one is improvement of quality characters. Say for example protein content of quality and reduction of toxic substances, and so and so. Quality character is also conferred by oligogenic character, so it is not an issue. The marker-assisted selection can easily transfer such a trait. Transferring desirable transgene once a transgene is transferred from one organism to another organism. Say for example BT gene is transferred from bacterium into cotton. The BT cotton can be used as a donor to transfer the BT gene into another cultivated variety, which is not PT. So transferring desirable transient can be possible with this. Next one is introgression of desirable genes from wild into cultivated genotypes. There are several traits which are favorable in wild type, which are not available in cultivated cultivars. So like disease resistance, insect resistance, fruit quality, and so and so. So this can also be transferred from wild type into cultivated ones. It is equally effective for both plants and animals. So there is no boundaries for marker-assisted selection applications of marker-assisted selection. So it can be applied to both plants and animals. Genetic improvements of tree species. In certain cases, the crop duration of tree species takes 20 years for mango. Say for example, for mango or several other tree crop species, it takes at least 10 years to 20 years for maturity. So selecting of plants at the seedling stage itself or the sap stage itself prior to maturity is a big advantage to conventional plant breeding program. Next, thank you.